Okay, good morning, everybody. It's really nice to see everybody here. And it was very nice of Martin and Helen. They kind of asked Paul and I to introduce and close the day. So I got to stand up and introduce. There are plenty of people here who can tell you all about volume electron microscopy. Today it's me. So I'm setting the scene, and I asked Martin and Helen if there was something particular they wanted me to talk about, and they said, no, go for it. So you've got some musings and introduce. So I thought I'd start off with a slide that I actually presented at the 3D EM, so the prior EM in situ structural biology community meeting, because the way the community has defined volume electron microscopy up till now is 3D ultrastructure through continuous volumes of greater than a micron, imaging resin-embedded cells and tissues. And even back when we defined this, there was a case for including cryo volume imaging as well. And I think that case is getting stronger and stronger. So it might be something that the community want to revisit. But I think the really interesting thing here is the continuous volumes greater than 1 micron. And when I was talking about this before, it was particularly in the context of in situ structural biology, where you're really limited to around 200 or 300 nanometers of depth by the penetration of the electron beam in the transmission electron microscope. So it's kind of interesting to think about why we need to image through continuous volumes. So there's a case for statistical sampling compensating for a lack of volume, and it's how we used to do things when we were only imaging in 2D in the transmission EM. So it's an old concept and it can be really useful. Stereology is the three-dimensional interpretation of two-dimensional cross-sections of materials and tissues. But there are plenty of cases for which stereology doesn't really hold in the kind of research that we do. And the original example for volume EM was from neuroscience. So this is the mind of the worm from Sidney Brenner's team, which took 10 years to map back in the 1980s. So here, serial section transmission electron microscopy was used. So for those of you who are electron microscopists in the room and who use ultramicrotomes, that was an effort that required thousands of serial sections picked up onto 3mm transmission EM grids and put into the electron microscope and imaged one by one. And if you lose more than a couple of sections, you lose your continuity of neurons and you have to start again. Of course, now things are different, and with automation in the microscopes and various other advances, we can do that automatically in around 3 weeks. So these are some really nice movies from Kedar Narayan's lab in the US, and they're available. You can click through the link on the volume electron microscopy community website if you want to use them. So there are 2 different flavors of volume electron microscopy. The first one, I think this video is the array tomography. So we have a load of cells grown on a glass cover slip or a piece of tissue or model organism. We prepare them for EM, embed them in resin, trim down the face to around 1mm squared, take a series of serial sections using now some automation in ultramicrotomes and put them down onto a flat substrate, and then image them one-to-one, create a stack of high-resolution images. And there's now automation in transmission electron microscopes that allows us to do the same kind of imaging across reels of tape. The idea is that eventually, and what today's meeting is all about, we can take that 3D data, we can model it, reconstruct it so that we can extract measurements which we can then compare in cells in health and disease, which sometime in the future will push what we do away from an imaging issue and towards a bioinformatics challenge. So the other group of techniques, those were array tomography style techniques. The other group are the block face techniques, where we start off in exactly the same way with cells or tissues. We fix them, stain them, embed them in resin, trim the block. But now, because we're going to a scanning electron microscope and imaging at the block face, you can fit the whole sample in. And not only that, you can fit either a miniaturized ultramicrotome with a diamond knife to do the cutting in situ. So a cutting, imaging, or we can put the block into a focused ion beam or a plasma system, which will automatically mill off very thin layers of material, much thinner than we can cut with a diamond knife. So again, we cut, image, cut, image. And these techniques are not only used in neuroscience, so they've spread out over the whole of the biological sciences. This is just an example from the Crick. So this is a zebrafish, and it's infected with toxoplasma parasites, and they're in green, and you can also see the macrophages in red. And so Marie-Charlotte Demart, my team, along with several research teams at Crick, used the light microscopy in fibritome slices to find the region of interest. So here's a red macrophage with 2 parasites inside in green. X-ray microscopy to help narrow down to the region of interest and to find the parasites. And then actually serial section TEM, serial block face SEM, and focused ion beam SEM, which is what you're seeing here. And continuity is important here as well. So these are the 2 parasites. And the features the team were looking for were breaks in the membranes, the host membrane that sit around the parasite, which indicates that the parasite is escaping from the kind of cellular immune response, and also the mitochondria that are sitting around the vacuole. So we need continuous volumes around a whole range of different questions. It could be neurons, it could be organelles. So we see whole-organism and correlative light and volume EM through neuroscience, through cell biology, plants and marine life, infection, and now being used on patient tissue as well. So what's next? So we are bound to see increases in speed, throughput, resolution, and scale in volume electron microscopy. In the 1980s, we were looking at the mind of the worm. So that's 1 millimeter long by around 100 microns in diameter. In the 2020s, we're looking at the mind of the fly. So 40 years later, and we have people here who are intimately involved in that, in those publications and in that process, and they can tell you more. And I usually show this figure because I think it's very powerful. So the future then is looking towards mammalian connectomes. So for the mind of the mouse, if the effort to map the mind of the worm is 1 airline seat in width, then the fly, the challenge of mapping the mind of the fly, 6.5 Boeing 747s, and the challenge of mapping the mind of the mouse is the distance from Boston to Lisbon. And that scale changes across the whole pipeline. So in sample preparation, in imaging, and in data analysis, again, which is why we're here today. So a team from the Crick and from the LNB, and Philippe is here today as well, were involved in this report for the Wellcome Trust, mapping the first mouse connectome and the effort involved. And in that report, if this is done by volume electron microscopy, and there is a chance that it'll be done by X-ray microscopy.
Electron microscopy.
Is that Alexa? Alexa, you might talk for me. Then if it's done by volume electron microscopy, then at the moment, the cost estimate, and it's not yet feasible, be $7.5 to $21.7 billion, which is very precise, and I can't even remember how we worked that out. We'll have to
play from current costs, right?
Yeah.
Which is always eyeballing it.
It's 0.7 that's very precise, isn't it?
I mean, at those ranges and those scales, right?
Yeah.
It's still 500 million.
Yeah. And you can find that report online. It's openly available. Just search Wellcome Trust and mouse connectome. And maybe of interest today, by far the biggest part of that cost is the proofreading of the image analysis. Down here we have an estimated cost of how much it would take to buy or create the microscopes needed, but the registration, segmentation, and the proofreading is enormous. And just for comparison, the cost adjusted for the Human Genome Project. So we always show this one as well. So in 2023, whole-organism was named as one of the 7 technologies to watch by Nature, alongside the James Webb Space Telescope and high precision radiocarbon dating. and advances in CRISPR. So all of that's very exciting. You have these big labs that are mapping brains, and then you have many of our smaller facilities like at the Crick, which are answering tens of big research projects on the smaller scale every year. And a lot of us are still doing this, and it's just not sustainable. So this is Anne in the lab. This is our data sets just on HeLa cells, and Anne is drawing around the nuclear envelope in every single image to make 3D reconstruction, which we can then start to look at the properties of and start to map. There have been huge advances. There have been huge advances in neuroscience in automatically segmenting neurons. There have been some advances in cell biology. So this is Kedar and Ryan's MitoNet. And that's applied to a FibSem data set from a HeLa cell. So it's working way better than a person could on this data set in terms of time. But there are still issues. So for those of you eagle-eyed people who are used to doing segmentation, some of these mitochondria are split into two, and some of them are joined with things that they shouldn't be. So actually, again, the proofreading and correction, even on the organelle scale, would be enormous. I tried to do some of this on the plane, but then I saved the file over itself and couldn't get the data back. So that also speaks to something else, which is we have a lot of developers in the field developing solutions for us. And I think whether you're developing hardware or software, you set a timescale for tool development, which I'm kind of convinced now is between 5 and 10 years, depending on what you're doing. And then you set a timescale for tool deployment. But actually what we're learning in our development is your tool deployment should be as long as the tool development to actually get it into the hands of end users who may not be able to code and may not even use a Jupyter notebook, even though Martin tries to train me. There are lots of types of software which are used in volume electron microscopy. I can't talk about all of them, but I think we're going to hear about some of them today and how CCP-volumeEM will help to support people to use them. Maybe just to say there's a whole world of image registration in X, Y, and Z that we still can't handle well, especially as our data that's coming in is getting bigger and we're stitching more. And then we have the whole correlation of multimodal data. So this is some work that we've been doing. So this is X-ray imaging to start with fluorescence microscopy layered over the top. And then whole-organism, you can see that the data sets have been registered using the nuclei as landmarks. And this is in human patient material from kidney transplant patients. So the red cell is an immune cell in a patient undergoing transplant rejection. The reason we can do this is some beautiful software from Christian Tisch's lab at EMBL in Heidelberg called MOBI. So we now have experts in the lab, but it takes a little while to get up and running with new software solutions. And something that's key for us here is also the OME-ZAR file format, which I think has largely been adopted by the community now, which is very important advance. And then we have Matthew here as well from Empire. We have deposited a lot of data at Empire in the Bioimage Archive. This is one of our first. It was the slide I happened to have ready. So this is a really critical thing for our community, which I think would be a very tight connection with CCP-volumeEM so that we can get all of our data up, not just so that other scientists can mine the same data and maybe use control data sets for what they're doing, which will help with maybe animal reduction in animal use as well. But also so anyone involved in AI can access these data sets. And there's still a chronic shortage of ground truth annotations for people who are developing AI. Luckily, we have an extremely strong whole-organism community. Again, this is an old slide now. So this was from back in 2019 when the communities first started coming together, seeded in the US by Kedar and in the UK with the Wellcome Trust, with Ardan, Herard, Paul, and I, and some of you were at that meeting. And then the online meetings during the pandemic, we had amazing co-chairs of the working groups in the whole-organism community. So we have websites, we have papers, we have training videos, we have all sorts of different resources. And that effort has been continued by led by Kirk, Michelle and Paul over the last few years with CZI funding. And with the video from Kirk, you can see the increase in the number of whole-organism facilities that have networked over the last few years. And I don't think this even goes up to where we are now. So we have an extremely well-networked community. We've had our second volume electron microscopy Gordon Research Conference. And just to flag, after the first whole-organism Technology Forum, which was hosted by Michelle, we'll have the second one next year in China, so please do sign up to that, where everybody can get together and really talk about the detail of what needs to be done technically for the community. And because the community is so strong, then we layer something like CCP-volumeEM on top of that, which should really help to bring the key people together to drive advances in how we handle all of this data that we're spitting out, so that we can either answer these big questions about connectomes, or we can support the researchers in our home institutes to add whole-organism data to all of the other omics and imaging that they're doing to answer their important research questions too. So with that, I would just like to thank the team that are running CCP-volumeEM. So Martin led the application, but we also have Michelle in the room and Matthew, and please stick your hands up and know if you're part of the team.
Mark.
Alan, yes, Martin, right, I haven't seen everybody yet. So thank you to all of you, because what you're doing is going to bring everybody together even closer and facilitate, I think, the next wave of whole-organism. And they're going to tell you all about that in the rest of the day.